Hello, welcome to another screencast where I'll be discussing the nucleic acids, the structure, and the function of them. So by the end of this, you should be able to identify and describe two examples of nucleic acids, be able to identify the monomers of the nucleic acids, identify three components of a nucleotide, describe how a polynucleotide is formed. So what is a polynucleotide and how it is formed. And then lastly here, discussing the relationship between a nucleotide with a T and a nucleoside with an S. And then what is the relationship between ATP and nucleosides there with an S. So let's get started. Let's start in the lower right here. Here's our DNA molecule in the double helix form. And this double helix is a very regular repeating pattern. And so it's coiled. And we have this major groove, and there's a minor groove here. The dimensions are very exact. You don't need to know these exact measurements here, but just to show you that the molecule is a very, very regular pattern. Right? And when this thing coils, you have this major and minor groove. That's going to be important later when we talk about regulation of some cellular processes here. So these are referred to as the major and the minor grooves. So if we unwind that double helix, we see that it's double because you have two sides, right? So I want to have this side right here, right? And then I have this side over here. The two sides of the molecule are bonded together by hydrogen bonds. This is important, right? That the hydrogen bonds are between these two sides, and they hold the two sides parallel to one another. Right? So between the thymines and the adenines, you have two hydrogen bonds. Between guanines and cytosines, between the G and the C, you have three hydrogen bonds. Right? The numbers aren't super important to remember, but there are some cases where it actually is. It is a factor. So the way I remember this is that GC3, GC3, these things all rhyme, so there's three hydrogen bonds here between the G's and the C's, and there's only then two between the thymines and the adenines. So T for two, there you go, and GC3 hydrogen bonds. So these two sides of the molecule are held parallel to one another by hydrogen bonds between the nitrogenous bases. Then the sides, as you remember from Bio 1, are made up of sugars and phosphates, a repeating sugar-phosphate backbone. So sugar, phosphate, right? Let's look at one of these strands here. And these strands, it turns out, that the sides of the molecule are actually polymers. This whole molecule is a polymer. So it's made up of individual monomer units that we call nucleotides. Okay? So really, DNA is a polymer. DNA is a polynucleotide, right? Because the nucleotides are the monomers, and you assemble those monomers together to get the polynucleotide. So polynucleotide, right, is an example of that is DNA. Right? DNA is a polynucleotide example. So let's go back to these nucleotides here. And the nucleotides are made up of subunits as well. Right? The nucleotides are, number one, phosphate groups. Number two, a pentose sugar, pent being 5. So this is a 5-carbon sugar, as well as then a nitrogenous base is the third component. So again, nucleotides you need to know are made up of phosphate groups, pentose sugars, and nitrogenous bases. You should be able to sketch a phosphate group here. You should be able to understand that this pentose sugar right here has 5 carbons in it, and that there are 2 different types of those, right, which we'll look at in just a second. And then there are nitrogen bases up in the upper left here that we'll talk about. So let's go talk about these pentose sugars here. There are 2 flavors of the pentose sugar. Each of them has 5 carbons, so in the green boxes down here. Right? So in DNA, it says it right here. So in DNA, you have a pentose sugar, and here are the carbons at the vertices, 1, 2, 3, 4, 5 up here off the ring. Right? So there's 5 carbons here, and they number them 1, 2, 1, 2, 3, 4, 5. Except I'm going to add something to this, right? They will put a little apostrophe here, right? And then apostrophe is a prime, so the apostrophe is read as a prime. So this is really the 1' carbon. This is the 2' carbon. Pardon me, 3' carbon, 4' carbon, 5' carbon. So if you're at the cocktail party and you overhear people talking about 1' carbon, 2' carbon, they're talking about either deoxyribose or ribose, they're talking about those sugars. If you hear them talking about carbon number 1, carbon number 2, they're probably just talking about a regular carbohydrate, right? So this prime is a special designation just to be talking about the sugars in the DNA molecule or the RNA molecule. So at the 1' carbon, if we compare deoxyribose to ribose here, because we're going to compare deoxyribose to ribose, let's just go carbon by carbon. So there's the 1', there's the 2', there's the 3', 4', 5', right? So if we look at the 1' carbon, no difference. Yeah. If we look at the 2' carbon, this has a hydrogen, and this one has a hydrogen, and oh, here's the difference right here, right? That at that 2' carbon, right, we have a hydroxyl group over here in the RNA, but over here in the DNA, we just have a hydrogen. So this is the difference. This is why this is deoxyribose, right? It is without an oxygen, right? And it's that 2' carbon. That's important. So how do you know if you're dealing with DNA or RNA? You look at the 2' carbon, and it'll tell you, okay? Another carbon that's important is this 3' carbon. So here's a 3' carbon, 3' carbon, hydrogen, OH, 3' carbon, hydrogen, OH. No difference right there, but this 3' carbon is going to be important in a little bit, okay? So knowing that that has a hydroxyl group on it there. Not distinguishing DNA from RNA, but just saying it's going to be important in just a little bit. So 4' carbon, no difference. 5' carbon, no difference between the two. So the only difference, again, is that 2' carbon. Right? There's the sugars. Let's go talk about these nitrogenous bases for a second. The nitrogenous bases up here are in 2 different families, right? 1 are the pyrimidines, and 2 are the purines. Here's what you do not need to know. You do not need to be able to sketch these structures here, right? You don't need to know those structures, but here's what you do need to know. You do need to know that the pyrimidines have a single ring structure to them, right? So these are single ring structures, and the purines then have a double ring structure to them, right? So pyrimidines are smaller, and the purines are larger. The way I remember which nitrogenous bases are which, the pyrimidines, right, pyrimidine has a Y, and cytosine has a Y, and thymine has a Y. So the ones that have a Y are pyrimidines. Now, I know you're sitting there, well, like, uracil doesn't have a Y in it. Yes, I know, but you remember from Bio 1 that when we're talking about RNA here, right, that there is no thymine here. So uracil replaces thymine, so thymine and uracil are kind of synonymous with one another. So you should just know that and then be able to tell me that uracil is a pyrimidine. Okay. Then the purines are not the ones that have Y, right? Adenine and guanine. Okay. So when you go back over to the structure over here, you notice that thymine has a Y, so that's a pyrimidine, and adenine doesn't have a Y in it, so that is a purine. And so we always have pyrimidine bonding with purine, right? Purine, pyrimidine, pyrimidine, purine. Always, always, always complementary, right? So the A is complementary to the T. So these guys are complementary to one another, and then the guanine, of course, is complementary to the cytosine. Right? Adenine would be complementary to uracil then as well. Okay, you just remember this from Bio 1. You memorized it. There you go. So the way this is a crazy way for me that I remember doing this in college for some strange reason, but it just stuck with me for the past 20 years, is that the way I was trying to remember which ones were the smaller ones and which ones are the larger ones was that I looked at this and I noticed that the pyrimidines were C and T, and I noticed that the purines were G and A, and I was like, oh, Connecticut, that's the abbreviation for Connecticut, and this is the abbreviation for Georgia, and Georgia is a larger state than Connecticut was, and that's just how I remembered it, right? Again, they're never going to ask you that on the AP exam, but just a way to maybe make things make sense, or I don't know, whatever. So there is the structure. Lots of information there. Now, this maybe seems like it's coming out of left field, but we had this ATP molecule earlier in the year, and now if you look at this, and this was on the free response that not a lot of people got, so let's take a second here, ATP, adenosine triphosphate. So this right here is the adenosine, and then here's the triphosphate part, so the triphosphate, and then here's the adenosine, right? So if we look at this adenosine, what is this? Well, this is ribose. Right? So that means on the 2' carbon you're going to have an OH, that's right. And since this is adenosine, this right here is an adenine nitrogenous base, right? So adenosine is adenine attached to a ribose, right? And this adenine, again, is a purine, and it's a larger one. Right? So if I dephosphorylate that, then I get adenosine diphosphate. Again, here's the adenosine part, right? And then there's the diphosphate part. If I take another phosphate off of there, right? So if I take adenosine diphosphate here and I take another phosphate off, as I'm showing you doing here, what do you get? Hello, you get this thing called adenosine monophosphate. Well, look at what this is actually. This is a sugar phosphate and a nitrogenous base, right? So AMP, this is really a nucleotide, okay? Now, you may come across this term nucleoside, right? And a nucleoside is something like adenosine. Right? So adenosine is a nucleoside. So really ATP is a nucleoside triphosphate. Right? A nucleotide includes the phosphates. A nucleoside does not include the phosphate. Nucleotide does include the phosphate group on there. Right? Minor difference, but it may trip some people up sometimes if you're reading or listening to another presentation. Okay? So we said that the DNA is a polymer. Right? DNA molecule is a polymer. And what we see here is that it's double-stranded, right? So there's a strand on this side and there's a strand on this side, right? And when the cell is making more DNA, like in the S phase, when we're replicating it, really what's happening is that you're taking these nucleoside triphosphates right here, right? And there is an enzyme that is actually going to catalyze this reaction. So now here's where that 3' comes in, right? This is the 3' carbon right there, and it has a hydroxyl group on it. This thing sticking up right here is the 5' carbon. And what we're going to do is to make this bond, making this bond is a positive delta G. And so the enzyme will break this bond right here. Right? Sorry, strike that. We'll break this bond right here. And when it breaks that bond, energy gets released. And when this breaking this bond is a negative delta G. And we're going to use that energy to then make this bond between the phosphate that's on the 5' carbon and the OH that's on the 3' carbon. I'll say that again. We're going to use this energy that's released from this catalysis here to make a bond between the phosphate that's on the 5' carbon and this OH group that's on that 3' carbon. This bond that's made, it's not a hydrogen bond. This bond that's made right here, right, is called a phosphodiester. A phosphodiester bond. Right? These bonds are covalent bonds between the nucleotides of the chain, of the side chain. So when we are polymerizing DNA, when we're making more DNA molecules, there's an enzyme that's going to add these nucleoside triphosphates to the 3' end. Right? So we add a nucleotide to the 3' end over here. That enzyme that makes phosphodiester bonds, that's making this DNA polymer, is called DNA polymerase. DNA polymerase makes phosphodiester bonds by adding nucleotides to, we say, the 3' carbon. Right? And it's always going to be lined up so that that 3' carbon is eventually then bound via a phosphodiester bond to that 5' carbon. Right? So you will, if you are doing this, so there's the 5', this is the 3' carbon, and then this would be the 5' carbon, and this would be the 3' carbon, right? And so on and so forth on down the line. Okay? So there is some detail about the molecule. There's some detail about how the difference between the different types of bonds. Here we see DNA compared with RNA. Right? So here is the DNA molecule. And I want to, again, emphasize here's the 5' carbon, here's the 3', and then you go 5', 3', 5', 3', 5', 3', 5', 3', 5'. That's why this is labeled as the 3' end. Right? Then this would be the 5' end. In the DNA molecule, the other side of the molecule, you notice here, is upside down, right? So that the 3' end is across from this 5' end here, and that this 5' end is across from this 3' end here. This relationship is important, right? And is described as being anti-parallel. So this is anti-parallel. The DNA molecule is anti-parallel. The sides go in opposite directions. Directions meaning 5' to 3', and then 5' to 3' is in the opposite direction. Right. So you might take a minute and go back and look at this, the sketch, or sketch this molecule here as to what it does not look like. Right. So what is it not? So maybe you sketch that over here. Right. This is what it looks like. It's anti-parallel. The two sides of that molecule is double helixed. Compared with an RNA molecule, which is only a single helix, right? So this is a single-stranded polymer, right? But you're still going to get this 5', 3', 5', 3', 5', 3'. So you still will have a 5' end referring to this carbon right there, and then the 3' end referring to that carbon right there. So if it's only single-stranded, right, you get RNA. Okay? And you notice that you don't have any T, you don't have any thymine here, right, which again is a pyrimidine, right, molecule. Okay, so there's a lot of information, and you should be able to now identify the two examples of nucleic acids, DNA and RNA, identify the monomers of those nucleic acids as nucleotides, be able to describe the three components of a nucleotide, and then how is the polynucleotide formed? Right? So we talked about that with the DNA polymerase and the nucleoside triphosphates. Right? And then the relationships between a nucleotide and a nucleoside, right, with or without the phosphate groups. And then how is ATP really kind of like a nucleoside? Okay? Thanks for listening. Summarize some stuff and bring your questions to class.